Hello, today we'll be talking about point mutations and the several effects they have at the protein level. First, let's start off with some key definitions. A point mutation is the substitution of one base for another. Purines are the nitrogenous bases adenine and guanine. Pyrimidines are cytosine, thymine, uracil. A specific type of point mutation is a transition, which is from purine to purine or vice versa. And lastly, there is transversion, which is from purine to pyrimidine or vice versa. Now going on to point mutation, first we have here is our DNA sequence. Before doing anything, it is important to remember that the mutation occurs in the DNA sequence. Now we need to transcribe this DNA into our mRNA sequence, and to do so, we need to change any thymines that we see into uracil. Each set of codons we see here codes for a different amino acid, and we can see that written down below each set. Now that we have our mRNA sequence, we will first talk about the missense effect. That is when a single base is changed and codes for another amino acid. Here we first had AAC, which codes for asparagine, and now changing that first A into a C, we now have the amino acid histidine. This change from adenine to cytosine is an example of transversion, and this change occurs in both the DNA and mRNA sequence as there is no uracil or thymine present. Moving on, we will talk about the nonsense effect, and that is when a base is changed and instead of being coded for an amino acid, it is now a stop codon. In this first case, we had UGC, which codes for cysteine, and now changing the C into an A, we now get a code for a stop codon. This TGA that we see here is the mutant DNA strand of TGC. This change from a C base to an A base is an example of transversion. Next, we have the silent effect, which is when a single base is changed, but even after the change, it still codes for the same amino acid. Here we have GCU, which codes for alanine, but changing that U into a C still codes for the same amino acid. This GCT that we see is the original DNA strand, and we change that T into a U. This change between U and C is a transition among two pyrimidines. Finally, we have the last effect, which is read-through. Read-through is when a base is changed and that causes the previous stop codon to be coded for an amino acid instead. In this example, we have UGA, which goes for a stop codon, but changing the A into a G, that allows for it now to code for the amino acid tryptophan. This TGG that we see is the mutant DNA strand of TGA. In that change from A to G, we see as an example of a transition between two purines. To recap, we went over the 4 different effects that point mutation have at the protein level, those being missense, nonsense, silent, and read-through. After watching this video, I hope you have a better understanding of DNA mutations, in this case specifically point mutations and their several effects on mRNA sequence. Thank you.